So now we will talk about protein. Protein is actually a polymer of amino acid, and amino acid can be written as A. So these are the basic structure of amino acid: carbon, hydrogen, oxygen, and nitrogen. Just now we mentioned that proteins are made up of polymer of amino acid. So now amino acid can be divided into 3 amino acids. So which is essential amino acid, semi-essential amino acid, and non-essential amino acid. So what is essential amino acid? Essential amino acid must obtain from diet and cannot be synthesized in human body. So what is non-essential amino acid? Non-essential amino acid can be synthesized in human body, so you don't need to obtain from the diet. These are the basic structure of amino acid. Amino acid consists of carboxyl group, COOH, which is acidic, and hydrogen atom, amine group, NH2, which is base, and amino acid radical, R, we also call side chain. So how to draw the structure of amino acid? NH2, this is the amine group. Then this is the H, and COOH, this is the carboxyl acidic group, and this is the side chain. Okay, now we move into protein classification. So protein can classify into complete protein and incomplete protein. Complete protein are all essential amino acids at the right ratio and quantity. So sources from complete proteins are from animals, which is eggs, milk, cheese, and meat. So incomplete proteins without one or more essential amino acids, mostly from plants, such as cereals, nuts, legumes, and seeds.
We can classify proteins based on their structure. First type is globular proteins. The second one is fibrous proteins. For globular proteins, the characteristic of globular proteins are water-soluble, compact, folded into spherical shapes, and globular proteins have complex tertiary and sometimes quaternary structures, and their roles in metabolic reactions. Examples are enzymes, hemoglobin in blood. For fibrous proteins, the characteristics are little or no tertiary structure, long parallel polypeptide chains, and fibrous proteins are usually insoluble. And lastly, fibrous proteins provide mechanical support. The examples are keratin in hair and collagen. Protein not only can classify by its structure, but it also can classify based on composition. First, simple protein. For example, albumin, lysozyme. And second, conjugated proteins. For example, hemoglobin, myoglobin, cytochromes, and lastly, immunoglobulins. Now we talk about source of proteins. There are two sources of proteins. One is from animal, which is good biological value, and for example, meat, poultry, fish, eggs, milk, and milk products. The other one is from plants. Like legumes can be found in soybeans, peanuts, cereal rice and wheat, and lastly, beans. Now I'm going to talk about zwitter ions. Under normal cellular conditions, amino acids are zwitter ions, which is also called dipolar ions. Main reasons are they have amino group, which is NH3 positive, and they have also carboxyl group, which is COO negative.
The function of proteins. They are the basic building blocks of living things. They are responsible for the growth and repair of body cells and tissues. They help with muscle contraction, muscle recovery, blood clotting, oxygen transport, third source of energy. However, some of the main functions carried out by proteins which are include. First is antibodies. They are specialized proteins involved in defending the body from antigens. They are traveled through the bloodstream and are utilized by the immune system to identify and defend against bacteria, viruses, and other foreign invaders. They are able to counteract antigens by immobilize them so they can be destroyed by white blood cells. Second is contractile proteins. They are responsible for movement, and these proteins are present in muscle contraction and movement. There are few examples of this protein which include actin and myosin. Third is enzymes. They are proteins that facilitate biochemical reaction and often referred to as catalysts because they speed out the chemical reaction. Examples of enzymes include the lactase and pepsin. Lactase can be used to break down the sugar lactose found in milk, and pepsin is a digestive enzyme that works in stomach to break down proteins in food. Fourth is hormonal proteins. They are messenger proteins which help to coordinate the certain bodily activities. Examples include insulin, oxytocin, and somatotrophin. Insulin regulates glucose metabolism by controlling the blood sugar concentration. Oxytocin stimulates contraction in females during childbirth, and somatotrophin is a growth hormone that stimulates protein production in muscle cells.
Fifth is structural proteins. They are fibrous and stringent and provide support. Examples include keratin, collagen, and elastin. Keratin strengthens protective covering such as hair, feathers, horns, and ribs. Collagen and elastin provide support for connective tissues such as tendons and ligaments. Sixth is storage proteins. They can be used to store amino acids. Examples include ovalbumin and casein. Ovalbumin is found in egg whites, and casein is a milk-based protein. Proteins also act as transport proteins. They are carrier proteins which move molecules from one place to another down the body. Examples include hemoglobin and cytochromes. Hemoglobin transports oxygen through the blood. Cytochromes operate in the electron transport chain as electron carrier proteins. Proteins also act as repair and maintenance. They are maintaining the body tissue, including development and repair. Children need more protein per pound of body weight than adults. The last one is protein also act as energy. Proteins are considered as major source of energy to living things. If you consume more protein than you need for body tissue maintenance and other necessary functions, your body will use it for energy. If it is not needed due to the sufficient intake of other energy sources such as carbohydrates, the protein will be used to create fat and become part of the fat cell.
The importance of proteins. It is a component of every cell in the body. In fact, hair and nails are mostly made of protein. Proteins help to build and repair tissue. Proteins used to make enzymes, hormones, and other body chemicals. It is an important building block of bones, muscles, cartilage, skin, and blood. Certain proteins serve as major structural role in tissues such as muscle and skin, and even provide the matrix for bones and teeth. Proteins are vital to cell division that is necessary for growth, reproduction, and healing. Proteins help to transport other nutrients around the body by binding to them and then releasing them when and where they are needed. Protein helps to regulate and maintain a proper fluid balance. This helps to maintain proper blood pressure and even lubricate eyes. Many proteins keep everything working right by regulating chemical reactions. Examples including enzymes, hormones, blood clotting substances, even receptors in the eyes. Protein is essential for the immune system to defend against foreign invaders such as bacteria and viruses.
We are going to discuss about protein structure. We got 4 types of protein structure. One is primary protein structure, secondary protein structure, tertiary protein structure, and quaternary protein structure. For primary structure, proteins are made out of polypeptide chains, which are amino acids joined together with peptide bonds. The unique sequence of amino acids that make up a protein or polypeptide chain is called the primary structure. In summary, primary structure is the unique sequence of amino acids that make up of a protein of polypeptide chain. Peptide bonds are created by enzyme catalyzed condensation reaction and broken down by enzyme catalyzed hydrolysis reaction. Breaking down protein is important in many areas of the body, not merely in digestion. For example, in hormone regulation, cells that are targeted by hormones contain enzymes to break down those hormones. This stops their effects from being permanent and allows them to be controlled. This is the picture for primary protein structure. We are made up of many amino acids to form a long polypeptide chain. For secondary structure, polypeptide chains are folded into different shapes, called their secondary structure. 2 common examples of secondary structure are alpha helix and beta sheets. Secondary structure is held together by many hydrogen bonds, overall giving the shape clear stability. In summary, secondary structure is the way in which the primary structure of polypeptide chain folds. This is the picture for alpha helix. We are held in place by hydrogen bond between every NH group and the oxygen of a CO group in the next turn of the helix. 4 amino acids down the chain. The typical alpha helix is about 11 amino acids long. For beta sheets, the beta sheet structure of the beta sheet is held together by hydrogen bonds between the amide groups of linear polypeptide chains. The average number of amino acid residues in a typical beta sheet is 6 with an average of 6 strands bonding together.
Now we'll continue to tertiary structure of protein. The final 3D structure of a protein is its tertiary structure, which pertains to the shaping of a secondary structure. This may involve coiling or pleating, often with straight chains of amino acid in between. Tertiary structure of protein is the final 3D structure of a protein, entailing the shaping of a secondary structure. Tertiary structure is held together by 4 different bonds and interactions. First, disulfide bonds, where 2 cytine amino acids are found together. A strong double bond SS is formed between the sulfur atom within the cytine monomers. Second, ionic bonds. If 2 orbitally charged R group, positive and negative, are found closer to each other, an ionic bond forms between them. Third, hydrogen bonds. Your typical everyday hydrogen bonds. And last, hydrophobic and hydrophilic interactions. Some amino acids may be hydrophobic while others are hydrophilic. In a water-based environment, a globular protein will orientate itself such that its hydrophobic parts are toward its center and its hydrophilic parts are toward its edge. Tertiary structure can be broken by the action of heat. Increasing the kinetic energy of protein with a tertiary structure makes it vibrate more, and so the bonds that maintain its shape, which are mainly weak, non-covalent bonds, will be more likely to break. When a protein loses its shape in this way, it is said to be denatured. Even when cooled, the protein will not form its original complex shape. Proteins with a 3D structure fall into 2 main types: globular and fibrous. Globular proteins tend to form ball-like structure where hydrophobic parts are towards the center and hydrophilic are towards the edge, which makes them water-soluble. They usually have metabolic roles.
For example, enzymes in all organisms, plasma proteins, and antibodies in mammals. Fibrous proteins form long fibers and mostly consist of repeated sequences of amino acids, which are insoluble in water. They usually have structural roles such as collagen in bone and cartilage, keratin in fingernails and hair. This is the diagram of the tertiary structure of protein, and the structure is held together by hydrogen bond, hydrophobic interaction, disulfide bridge, and ionic bond.
Now we want to talk about the quaternary structure. Some proteins are made out of multiple polypeptide chains, sometimes with an inorganic component. For example, a heme group in hemoglobin, called a prosthetic group. This protein will only be able to function if all subunits are present. The quaternary structure formed when two or more polypeptide chains join together, sometimes with an inorganic component, to form a protein. This is a diagram for the quaternary structure. Hemoglobin and collagen. Hemoglobin is a water-soluble globular protein which is composed of two alpha polypeptide chains, two beta polypeptide chains, and an inorganic prosthetic heme group. Its function is to carry oxygen around in the blood, and it is facilitated in doing so by the presence of the heme group, which contains a iron 2 positive ion, onto which the oxygen molecules can bind. The collagen is a fibrous protein consisting of three polypeptide chains wound around each other. Each of the three chains is a coil itself. Hydrogen bonds form between these coils, which are around 1,000 amino acids in length, which give the structural strength. This is important given collagen's role as structural protein. This strength is increased by the fact that collagen molecules form further chains with other collagen molecules and form covalent cross-links with each other, which are staggered along the molecules to further increase stability. Collagen molecules wrapped around each other form collagen fibrils, which themselves form collagen fibers. The collagen has many functions. First, form the structure of bone. Secondly, make out cartilage and connective tissue. Third, prevents blood that is being pumped at high pressure from bursting the walls of arteries. And the last is the main component of tendons, which connect skeletal muscles to bone. Then the hemoglobin may be compared with collagen as such. As we said, hemoglobin is globular while collagen is fibrous. Solubility. Hemoglobin is soluble in water while collagen is insoluble. Amino acid constituents. Hemoglobin contains a wide range of amino acids, while collagen has 35% of its primary structure made out of glycine. Prosthetic group. Hemoglobin contains a heme prosthetic group, while collagen doesn't possess a prosthetic group. Tertiary structure. Much of the hemoglobin molecule is wound into alpha helix, while much of the collagen molecule is made out of left-hander helix structure.